Hello everyone. We are going to try to use this SAC similarity tool. I'm Greg Cura and I helped develop this with the support from Kevin Dyer. It's a JavaScript and it helps you analyze data collected in high throughput. The files I'm going to be working with are available at the bottom of this website. Again, the website's Sybil's SAC similarity. Going to the bottom, we have this file called morph.zip, and in morph.zip we have a bunch of SACS profiles calculated from this morph. So every step, there's 21 steps going from open to closed. I've randomized those files, so I've give it random number 00, which is frame number 2 from this morph. Random 01 is frame number 16, etc. And I've done this for demonstration purposes, as you'll see. So this is a drag and drop tool. And what one can do here is take the files from inside there in two ways. You can drag and drop just like this, or select and then drag it, or you can click and select them. And this is actually the safest way. And then hit open. And these should load pretty quickly depending on your internet speed. So these guys are loading up here. And once all 21 frames are done, you should see a heat map. And within this heat map, what we're doing is comparing pairs of SACS data profiles. What's happened is randomized frame number 1 is compared to randomized frame number 2 here, and because they're actually close in terms of real frames, 15 and 16, they're pretty similar. And as you go across, you can see where the most similar and dissimilar they are. If you were to click on one of these boxes like this, the box will now have the delta RG that it measures for that comparison of randomized frame calculated scattering profile 1, the same for randomized 2. It will also show you the scattering profile down here. Now, if you have certain specific regions that you're particularly interested in, or your data gets cut off in some way, you can adjust what regions are of interest. So let's say we were just interested in sort of the bumpy parts of this curve here. This adjusts what parts of the curve are being compared against one another. The default somewhere in the point, well, I have data to 0.01, but you can extend it to 0.24, etc. I'll go to 0.2 here. You can also try to create similarity. So, frame 15, 16, you can bring this red frame number 11, you can drag it closer, and it will rearrange the whole matrix. You can choose what you use for judging similarity between scattering profiles. So the traditional one used in SACS is chi-squared. We have found that measure to be not the best in our hands. We like volatility of ratio, which is something we've developed. There's also a Pearson correlation coefficient and something developed by Sokolova et al. And you can pick that one as well. And again, these should generally show similar qualitative trends. Going back to volatility of ratio, you can also adjust the color spectrum that you're using. The blue, white, red to me is sort of a 3 color system where you can say agree, not agree, sort of agree. The yellow and red is colorful. You kind of have a sense as it goes from red, orange, yellow, white. There's a gradient. The more true one to get that sort of gradient color is the black to white, of course, and that's a classic as well. There's a diagonal that's symmetric here, and that's because that's comparing the same to the same, so those aren't scored. So going back to red and white, this one here is comparing randomized frame 7 to randomized frame 7, so that was a perfect score, and I didn't want that to change the contrast, count as part of the contrast. Sometimes you'll end up with scattering these maps where there's just one white row. That means that scattering profile is so far different from everything else, and you may consider getting rid of that in your next reload of this tool, because it's sort of adjusting the contrast based on that. Another operation, and this is why I've done this randomized thing, is to use this clustering option. When I hit cluster, it should group those that are similar together and those that are dissimilar far apart. And when I do that, that's what happens. And you'll see that frame 0 is at the very bottom, frame 1920 is at the top. We can adjust that slightly. And what this has done is, based on scattering profile, ordered the morph. It knows that similar scattering profiles have similar structures. Kind of the point of this web app is that it's can help you with this model. So if I click on these very dissimilar curves, this frame 0, random 14, and compare it with the very first one, frame 20 here, you can see these profiles are very different, and they correspond with the first and the last part of that morph. Also, you'll see that the radius gyrations are described here, and the delta RGs are described here. Another very valuable tool is this force plot. If you click force plot here, it will change how this is viewed. Each scattering profile becomes a node. I'll click it here. And the force of opposition between one node and the other is set by the similarity score. So this frame 0, randomized frame 14, is very similarized to frame 1, and you can see how you basically get that trajectory as well. And the most dissimilar, the ones that have the largest force of opposition, are the first and last frame. So again, you clearly see the relationship between everything. So you can go back to the heat map here. You can create a matrix, convert this to an image, let's put it that way. So this will create an image that you could work off of for a presentation or for whatever you need. You can show all the RGs along the whole plot and plot that out. You can also output the VESA bar scores here, how this is all scored out, and save that to a file. You can do that for chi-squared, for example, and you can hit the force plots. You can see how that works out for you. You can also do the Pearson and the Sokolova method. So again, how all these things change, and you can print this image as well. What I like to do, if I'm going to put this in a publication, is I like to copy this image and then dump it into like a Photoshop or something like that, and copy the circles. It takes a little while, but again, you can get a lot of information this way. So back to heat map. You can pull down another data set at the bottom here. Oh, there's that image. You can pull down an experiment we did on this same system, and this is a salt versus pH experiment. So I'm going to pull that down, That'll get dropped into my downloads folder here, and I can reload this guy and take my experimental data. Here. Bring that on. While that's loading, I can quickly also load up these morph results. Oh, wasn't quick enough. Let's try that again. That time I got it. And what you're going to be looking at is at this real construct, the one we've done an experiment on, in the context of these this morph. And what we've done is this triple mutant at salt of 500 millimolar pH 4, salt of 10 millimolar pH 7. We're looking at here a lot of scattering curves and a lot of experimental data together, and we're seeing how close the experimental data matches our model. So now if I go to this, and there is information here, but it would take a little bit of time to extract that. If you go to the force plot, I like this version much better. It more quickly tells you what's happening. You can drag and drop the system to orient it a bit. You can see our most closed morph, and I did not mention this before, but I should have. That is that the size of the circles are proportional to the raised duration. So you can see that this most closed, smallest conformation, this calculated model, is actually very close to the triple mutant at a 10 millimolar salt and pH 7. And then you can go through this data set and you can see what affects opening and closing. So as you increase your salt to 500 millimolar salt, you're heading towards the open structure. The pH is roughly the same, so it looks like salt opens the structure. Going from top to bottom, you see there's a low pH here and the high pH up here. So salt drives it across, pH drives it up and down. What's occurring in the system is that the cage, this cage system, sorry, I probably shouldn't have done that. This cage system here is disassembling and sort of forming aggregates and things like that as you go from low to high pH. And that's all I wanted to really share with you guys. I hope you learned stuff, and please, if you use it, to cite a Nature Methods paper that we use to announce this result. Thanks for your time.